Let's review the 5 different types of eukaryotic gene regulation. These are some pretty difficult concepts to wrap your head around, and for AP Biology, I really only have the students understand generally what's going on at each level. The 5 main types of gene regulation that we're going to talk about are chromosome, transcriptional, post-transcriptional, translational, and post-translational. Our first type is the chromosome structure, and this is going to regulate how RNA polymerase is going to bind to a certain gene. Remember that chromosomes are tightly packed structures of DNA and protein. Specifically, the protein is called a histone that the DNA is going to wrap itself around. Whenever methyl groups are added to histones, the DNA is going to tightly pack around those histones, not allowing RNA polymerase to bind and transcribe a gene. Whenever we add the acetyl groups to the histones, it's going to loosen the DNA. This allows RNA polymerase to bind more easily and transcribe a gene. Next, let's talk about transcriptional control. This is going to be the most important type of regulation for eukaryotes, because if we stop transcription, we stop translation, which will be more effective. Transcriptional factors are going to regulate how much RNA polymerase is going to produce mRNA. Activators are going to be transcriptional factors that activate transcription, while repressors do the exact opposite. They're going to stop transcription. After mRNA is produced, it's going to go through something called post-transcriptional control. This type of control looks at how the mRNA is going to function after it's produced. And there are many different factors that are going to influence post-transcriptional control, such as alternative splicing, RNA editing, and RNA degradation. Basically, these all influence how mRNA is going to function within the cell. Our next type of control is translational control, and this influences how much the mRNA is translated at the ribosome. This can be like the lifespan of mRNA, which is usually longer for eukaryotes and shorter for prokaryotes. And this is because the prokaryotes need to respond to their environment much quicker. And also something called RNAi, which I'm not going to get into this TikTok because there is so much that RNAi can do. But basically, RNAi interferes with how much RNA is going to be translated. Our last is post-translational control. This occurs after the protein has been produced. Proteins can be modified through their structure and function, and these are usually done by things like chaperone proteins. Or proteins can be tagged with a molecule that signal them to be broken down, because once those proteins aren't needed anymore, the cell can use those amino acids for other functions. So you can see with our molecule here that's tagged, it's going to be eventually broken down by a structure known as a proteasome. And then these polypeptides and amino acids will become other proteins, almost like a recycling center.